Today we'll discuss about transcriptional regulation of gene expression in eukaryotes, right? There are two kind of organisms, prokaryotes and eukaryotes. Prokaryotes include bacteria and blue green algae, and their regulation of gene expression we have discussed in detail in previous videos. A link will be shared in the description box. But in this video we'll discuss about regulation of gene expression in eukaryotes. Now, this to explain the working of genes in eukaryotes, a model was proposed by Britton and Davidson in 1969, and this model has been named as gene battery model. Why it is called as gene battery model? First, we'll discuss different kind of genes present here. Then we'll discuss why it is called as gene battery model. Now, this comprises of four genes: producer or structural gene, receptor or operator genes, integrator or regulator gene, and sensor gene. And if we see in the prokaryotes, now these are called as structural genes, operator genes, and regulator genes. But in case of eukaryote, it is producer, receptor, integrator, and sensor genes. Now how these genes they work? First we'll discuss in detail what are producer or structural genes. Basically, these genes are involved in the transcription activity. So, what is transcription? Formation of RNA from DNA is called as transcription. Now, these structural genes they will be identified by RNA polymerase, and then they help in formation of RNA, and this RNA later will be converted into protein by translation process, and genes will be expressed. Now, how this structural gene is being regulated, that we will discuss. So, these genes which help in formation of RNA and proteins, and they are involved in various metabolic pathways present on the DNA, may be present on the same or different chromosomes. Now, the second kind of gene are receptor or operator genes. Now, this receptor or operator gene is linked with the structural gene. We can see here. Now, this will receive the information, operator gene or receptor gene. This will receive the recognized by RNA polymerase. Then RNA polymerase will move to the structural gene for the transcription process. And every structural gene which has its own operator or receptor gene, or we can say स्ट्रक्चरल जीन को चलाने के लिए क्या चाहिए, ऑपरेटर और रिसेप्टर जीन. रिसेप्टर का मतलब होता है रिसीव करना. So each structural gene has its own operator or receptor gene, and which is lying adjacent to the structural gene. So basically, if this is operative, then only RNA will be produced or transcription will occur. And if this is not operative, then RNA will not be produced, and later on protein will not be produced. So receptor or operator gene is very important for the functioning of the structural gene. Now, third gene in this model is integrator or regulator gene. किसी भी चीज़ को कंट्रोल करने के लिए उसकी रेगुलेशन इज़ वेरी वेरी इंपॉर्टेंट. So that can be done with the help of regulator gene. Basically, this regulator gene or integrator gene, this also produces an RNA. Now, this RNA is not for protein synthesis. This is for a regulatory function, and we also call it as active RNA. Now, this will control the operator or the receptor gene, and then only structural gene will undergo transcription. So, integrator or regulator gene provides an activator RNA which will promote the functioning of operator gene directly or through the production of the proteins. So, these are called as integrator or regulator gene. Now, fourth kind of genes are sensor genes. As the name indicates, something is being sensed. So, this is called as sensor gene. Gene को काम करना है नहीं करना है, that will be sensed by the sensor gene. What are the environmental factors inside the cell? That is called as intracellular environment. Whether there are certain hormones, vitamins, chemicals, certain metabolic ions, or certain bacteria, viruses, or microorganisms. So those are going to control integrator or regulator gene. If this gene has sensed that this integrator regulator gene should work, then it will work. If sensor gene has sensed it should not work, then it will not work. So this is about sensor genes. So every sensor gene will control the integrator or regulator gene, and then only transcription will take place and metabolic activities will occur. So some of the sensor gene may act as transcription factors also. Like transcription factors are the factors which control the transcription process. Now, if transcription factors are active, then only sensor gene will work, and if transcription factors are not active, then this sensor gene will not work, and if sensor gene is not working, then integrator or regulator gene will not participate in the formation of RNA and later on proteins. So there are four kinds of gene as we discussed: producer or structural gene, receptor or operator gene, integrator or regulator gene, and sensor genes. So we can see here one gene is being controlled by other genes. So we can see a set of structural genes controlled by one sensor site is termed as battery. Now, one sensor site may be associated with many integrator or regulatory gene, causing transcription of all the integrators at the same time. This is regulating the producer gene through the receptor site for the production of RNA which is required for the protein synthesis. And for the functioning of gene battery or for the functioning of these genes, sometime their activity is enhanced with the help of enhancer gene. So enhancer gene will increase the transcription activity, that is, they will increase the formation of the protein. And if it has to be stopped, then this is done with the help of silencer gene, which will stop the activity of the gene, and they are regulating this gene battery model. So we can explain in detail like here. Like this is the sensor gene which is associated with the integrator or regulator gene. Now this regulatory gene, when it is being sensed, either hormones are there or certain vitamin minerals are there, pathogens are there, or we have certain transcription factors which act for the sensor gene, then integrator or regulator gene will help in production of the RNA, undergo transcription. Now this is called as activator RNA. This activator RNA will be received by receptor gene. Now this receptor gene will receive this RNA. This will be regulated. This gene will be regulated. Then this will move over to the structural genes, and structural gene will undergo transcription process to form the messenger RNA. And later on, this messenger RNA will undergo translation process. What is translation? Formation of protein from RNA is called as translation. And later on, this messenger RNA, with the help of other RNAs, will help in the production of the protein. And those protein, they may be involved in many metabolic pathways. Now we can see here sensor gene is regulating the integrator or the regulator genes, right? So producer or integrator genes are the sequences which are involved in the RNA synthesis. Receptor and sensor genes are those sequences which help only in recognition without taking part in RNA synthesis. They are not doing RNA synthesis, but they help in RNA synthesis. So Britton and Davidson also proposed that receptor sites and integrator genes may be repeated a number of times, right? So receptor site, this is receptor site, and this is the integrator site. इनकी रेपिटेशन बहुत ज़्यादा होती है ताकि ये बाकी जीन्स को कंट्रोल कर सकें. Repetition of receptor site ensures that same activator will recognize all these genes and several enzymes of one pathway which are being simultaneously synthesized. So this also explains that there is a repetitive DNA in eukaryotes. So the repetition of integrator and receptor genes will help in that repetitive DNA to function. So this is all about regulation of gene expression in eukaryotes. Thank you for watching my video. If you have any questions, please ask me in the comment box. If you like my video, please like, share and subscribe.